Welcome back again to the class of biology, and we will continue with the lesson the cell, and we were doing the cell organelles, and we'll continue with the cell organelles. We'll start with the flagella and cilia. Ribosomes are left. We'll continue ribosomes after flagella and cilia. So flagella and cilia, these are the locomotory organs of certain organisms, right? So they are fine hair-like structures. They are movable protoplasmic processes that are present on the cells, which are capable of producing a current in the fluid, right? Fluid medium for locomotion or even for passage of substances, right? So that is what the flagella and the cilia are used for, right? So they could be locomotory organs also, and they could also be used for movement of certain substances. Flagella are generally longer as compared to the cilia, and they are used for motility also, right? Cilia are also present. Cilia may be present all over the body also, right? And they are present in certain structures, like they are present even in our respiratory organs, right, to just throw out the dust particles that may enter, right? So it protects it from entering it protects the lungs from the dust particles, so it does not allow the dust particles to enter, right? So just before we talk about the differences between cilia and flagella, let us first understand the structure, right? So both cilia and flagella are structurally similar, right? What difference we know is that cilia are smaller in size and the flagella are longer in size, right? So basically they are similar in structure and possess the same parts, right? So what are the parts of these locomotory structures, or let's say the flagella and the cilia? They are the basal body. If you look at this, the basal granule that is labeled over here, that is the basal body. Then you can see the rootlets that are there, and the basal plate here. This is the basal plate, and this long structure is the shaft, right? Shaft is the structure that is visible in the form of the flagella or cilia. The basal plate, and beneath that they are all embedded inside, right? So they are attached to the to the protoplasm, and some part of it is in the cytoplasm of the cell, right? So let us start with the basal body. Here, this part, the basal body, it is also known as kinetosome, right? So it is called the basal granule, as I have labeled here. This part here is the basal granule or the basal body, right? Also known as kinetosome. Now this part occurs embedded in the outer part of the cytoplasm, right? So if this is the cytoplasm, then this is the outer part of the cytoplasm. The whole thing is the cytoplasm. This is the outer part, the part that is near the cell membrane, right? So it occurs embedded in the outer part of the cytoplasm below the plasma membrane. Now for example, if I draw this as the plasma membrane, right, and the inner part, inner to the plasma membrane, this is the cytoplasmic layer, right? So somewhere here you will find the basal plate, the basal body, right, or the basal granule, right? So it is like a micro cylinder, cylindrical in structure, microscopic, right? So it is like a micro cylinder which has a structure that is similar to a centriole. Here, this is the centriole. We will talk about the centrioles also after we finish the flagella and the cilia. Now this part has triplet fibrils we can say, right? It has triplet fibrils which are present on the periphery. What is the periphery? The side, right? So triplet fibrils which are present towards the periphery without the central fibril. Now look at the structure here, the part that I have drawn in green. This is the fibril, and if you look at the fibrils closely that I have drawn, you will see that I have drawn 3 strands here, which I have labeled the innermost as subfibril A, then the middle one is the subfibril B, and the outermost is the subfibril 3, right? So it's a triplet fibril, 3 fibrils found together on either side, right? Towards the periphery, let's say, right? So that is the triplet fibril. Innermost is the subfibril A, the middle one is the subfibril B, and the outermost is the subfibril 3, right? So that is the triplet fibril present on the periphery without a central fibril. Now if you look at the shaft part of the flagellum, you will find this blue, the diagram drawn in blue, right? This blue part is the central fibril that is present, right? Now if you look at the central fibril, you will see that it is present only in the shaft region, and the central fibril is absent in the basal granule region, right? So that means the basal granule or the basal body is devoid of the central fibril, right? Devoid means the central fibril is not present, right? And we find a hub of protein present over here, right? So if you look at these 2 black lines which I have labeled as hub, right? These black lines are we can say an aggregation of protein, right? It is a hub of protein, right? So what do we find in the basal granule? First of all, the basal granule part is present inner to the plasma membrane and slightly towards the outer side of the cytoplasm, right? It is embedded in that, right? What is present in the basal granule of the basal body? This is known as cylinder, right? Cylindrical in shape, microscopic, right? We find triplet fibers towards the periphery. What are triplet fibers? 3 fibrils that are present, right? Apart from the triplet fibers, we find a hub of protein, right? And there is no central fibril present in the basal granule, right? So that's the basal body. If we move lower down, you can see some root-like structures which are drawn in red ink, right? Root-like structures, and they are labeled as rootlets, right? So these are striated, right? Striations are there, right? Fibrillar outgrowths. Fibrillar made up of fibers, fibrils, microscopic outgrowths coming out from the basal body, which develop from the outer lower part of the basal body, right? If you look at this, these are, if you look at the rootlets are arising from the basal body. This is the upper part of the basal body. Here this is the lower part of the basal body, and we see that the rootlets, they arise from the lower part of the basal body, right? What is the function? They are meant for providing support to the basal body, right? So the rootlets are made up of bundles of microfilaments, right? Bundles, they are microscopic, but they are bundles of microfilaments that makes up one single rootlet, right? So they are meant for providing support to the basal body, right? So we've talked about the basal body and the rootlet in the flagella or the cilia, because the structures are almost the same, right? So we move on to the next part of the flagellum, that is the basal plate. Here, thick transverse line drawn in black ink, right? So this is the basal plate. It is an area of high density, right, which lies above the basal body. Now this is the basal body, this is the upper part of the basal body, right? So the basal plate lies above the basal body and it is high in density, right? And this is found at the level of the plasma membrane. That means that this basal plate is present somewhere over here at the level of the plasma membrane, right? Now in the region of the basal plate, this is the region of the basal plate which is found at the level of the plasma membrane, we find there are one subfiber of each peripheral fibril disappears. Now look at this, the outermost fibril, right, the outermost fibril of the triplet fibril, the outermost one is missing near the basal plate region, right? That means the triplet fiber, out of the triplet fibrils, the outermost, that is the subfibril C, is found only till below the basal plate. At the basal plate level, subfibril C is missing, right? So it disappears. The central fibrils, here you look at this, the basal plate, the central fibrils, they start from the basal plate area, right? Next part above the basal plate is the shaft, and this part is the actual flagellum or the cilia which are visible, right? So shaft, it is the hair-like projecting part of the flagellum or the cilium, right? Flagella is the plural of flagellum and cilia is the plural of cilium, right? So this is the outermost hair-like part that is visible. The length is 5 to 20 μm in the case of cilium, and flagella are more longer in length, so around 100 to 200 μm, right? Now if we look at this, we find that the shaft is covered on the outside by a sheath which is extension of plasma membrane, right? It is covered. If you look at this black portion, this is the flagellar sheath, right? So it is covered by sheath, right? Internally, that means inside the sheath, what is present? It contains a semi-fluid matrix. Semi-fluid makes it movable, right? So it contains a semi-fluid matrix having an axoneme of 9 peripheral doublet fibrils and 2 central singlet fibrils, right? So 9 peripheral doublet fibrils. Yes, periphery is towards the side, right? So this is towards the side. 9 peripheral doublet fibrils. If you look at these 2 green fibrils, they are 2 fibrils towards the side, right? Like this, there are 9 sets, right? 9 peripheral doublet fibrils all towards the side, right? This is doublet 1, 2, 3, 4, 5, similarly 9, right? All doublets made up of 2 fibrils, right? So that is the axoneme with a semi-fluid matrix. Towards the periphery side, 9 peripheral doublet fibrils, right? Other than that, if you look at the central part in blue, it contains of 2 central singlet fibrils, 1 and 2 here. 2 central singlet, blue ones are single, one on the side and one on the other side, right? 2 central singlet fibrils, right? Now this arrangement is known as 9+2 arrangement, right? This arrangement of the fibril in the shaft is known as 9+2 arrangement, or also known as 11 stranded, right? 9+2 arrangement, whereas in the centriole, where we will see, it is only 9 plus 0 arrangement, right? We will only find that there are 9 doublet fibrils that are present, right? So that's the arrangement in the centrioles. Now and the basal body, of course. The 2 central singlet fibrils, if you look at these 2 central singlet fibrils, they are covered by a proteinaceous central sheath, right? If you look at these oblique lines are a sheath or a covering, right? So that means the 2 central singlet fibrils, they are covered by a central sheath which is made up of protein, right? So that is proteinaceous, right? So the different proteins that make up the all the coverings, right? All the sheaths. So 9 plus 2 arrangement of the fibrils in the flagellum, right? Now the function, as we know that their function is, we can say, locomotion, right? They are motile, the flagellum and the cilia are motile, their function is locomotion, and if it is not locomotion, then they are present in many cells which, you know, throw away certain particles that are not required, right? So cilia and flagella, they move by sliding of the doublet fibrils against one another. See how it slides, right? It will slide like this over one another. What are the doublet fibrils? Here towards the periphery. These are the doublet fibrils, right? Now for this, of course, energy is required. So energy provided by ATP is used. Now flagella perform independent undulatory movements, right? Now what is the movement of the flagella? Undulatory, like how the snake moves, you know, like this. Undulatory movement. This is the movement of the flagella, right? So let's quickly go through the differences between cilia and flagella. We find that the number of cilia per cell is usually very large as compared to the number of flagella. So if we talk about the number of cilia per cell, it may be from 300 cilia to around 14,000 cilia, right? So that is the number of cilia present on the cell. If you remember paramecium, the paramecium is covered by the cilia all on the outer surface, right? The number of flagella per cell is usually 1 to 4, right? Cilia are much smaller. We discussed about the size as compared to the flagella, which are longer, right? And cilia, if are present, it occurs throughout the body or the major part of the surface of the cell, whereas flagella are commonly found at one end of the cell. So if you remember the bacterial cell, you see where is the flagellum present? It is present only towards one end of the cell, right? In cilia, the beat, that is the movement, is a coordinated movement, right? In a coordination, in a rhythm, they move, you know, all together. They'll move at one side, right? Or they may move one after the other also. So it is all rhythmic movement, whereas the flagella, they beat independently, right? Beat means the movement. The cilia produce a sweeping or a pendular stroke, like this, pendular stroke, sweeping or a pendular stroke, right? How do you sweep? Like this. Pendular stroke, how does the pendulum move? That is the kind of movement of a cilia. Flagella produces undulatory movement, I told you, like how the snake moves, right? Wave-like movements. Cilia help in locomotion, it helps in aeration, it helps in feeding, it helps in circulation. So wherever the cilia are present, right? Flagella basically helps in locomotion only, right? Cilia may fuse to form a membrane, whereas flagella, such kind of fusion with any membrane is not known, right? The surface is smooth in a cilia, whereas surface may be smooth or may possess some flimmers, right, over the surface. The function, as we've already discussed, is basically locomotion. They also create current for obtaining food from aquatic medium. It is also called the food current, right? In some protists and in some animals, the organelles take part in capturing the food also, right? Then internal transport of several organs is carried out by cilia, performed by cilia. For example, the passage of eggs in the oviduct, right? The movement of eggs in the oviduct. Then the passage of excretory substances in the kidneys, etc. is performed by the presence of cilia over the surface of the cell, right? So they are sensitive, the cilia and flagella are sensitive towards light, temperature, contact, etc. Right? So those are the functions of the cilia and the flagellum. And we move on to the next organelle, which are the centrioles, right? So centrioles present only in the animal cell, and when we talk about centrioles, we see that these are minute, very small, submicroscopic, microtubular, microscopic, tube-like, microtubular subcylinders, right, with a configuration of 9 triplet fibrils, right, and ability to perform their own duplicates. They can form their own duplicates, right? That is the ability, right? They have astral poles, basal bodies, all these things they can form without having DNA, right? They can duplicate in the absence of DNA. That means they don't have DNA and a membranous covering, right? So it says here they are approximately 0.3 to 0.5 μm in length and 0.15 μm in diameter. So microscopic, they are visible under the light microscope, but the details of the centriole structure were revealed only under the electron microscope. So two centrioles are found associated together, and both the centrioles are located at right angles to each other, right? So the pair of two centrioles are known as diplosome, and this diplosome, it lies in a common specialized part of the cytoplasm, and that specialized part of the cytoplasm is known as the centrosphere. Now in this centrosphere region where these centrioles are present, there are no other cell organelles present. So it is devoid of all the other cell organelles, right? It however may contain a few fibrous materials, and the complex, the centrosphere along with the centriole together is known as the centrosome, right? So Boveri in 1888 called it the centrosome, right? Boveri in 1888. So in 1888 Boveri called it the centrosome. So what are the centrosomes? It is a complex structure of the centrospheres along with a pair of centrioles which are placed at right angles to each other, right? So that is the centrosome, right? They are found, these centrioles are found in all the eukaryotic animal cells, right? Then they may be found in certain protists also, except the amoeba. They are capable of replication, as I told you, duplication. So they are capable of replication, and centriole replication is coordinated in animal cells with cell division. So when do they replicate? At the time of cell division, right? So it occurs in, when we will talk about the cell division, the mitosis and meiosis, then you will study there is one phase S or G2 phase. So in that phase it starts dividing, right? So this is prior to the nuclear division. The two centrosomes, they separate and they move towards the opposite poles, right? So where spindle fiber formation starts taking place. So when we will do cell division in detail, then you will understand the role of centrioles, right? But for now you must know that centrioles are the cell organelles which are present in the animal cells, right? So centriole replication also occurs at the time of the formation of basal bodies of cilia and flagella, right? At that time also, right? So what is the structure like? It possesses a whorl of 9 peripheral fibrils, 9 peripheral fibrils. Fibrils are absent in the center. So it is 9+0 arrangement, right? Remember, flagellum we talked about the 9+2 arrangement, and here the arrangement we call is 9+0. Why? Because 9 peripheral fibrils are there, but in the center there is no fibril, right? So 9+0 arrangement. The fibrils they run parallel to one another at an angle of 40 degree. Each fibril is made up of subfibrils, right? So therefore it is called triplet fibril. The 3 subfibers are in reality microtubules, and they are joined together by their margins, therefore sharing a common wall, which is made up of 2 to 3 protofilaments also, right? So again, the subfibrils, if we name them, if we label them, it would be subfibril A, B and C, right? So 3 subfibrils, triplet, and 9 triplet subfibrils towards the periphery without the presence of the central fibril. So the arrangement is the 9+2 arrangement, right? Now what is the function of the centrioles? The centrioles have not been found to contain DNA. We've already discussed that. Even without the presence of DNA, they can replicate or they can duplicate, right? Yet they are capable of forming new centrioles with the help of certain we can say proteinaceous structures, etc., that are present, right? Then centrioles help in cell division by forming spindle poles or microtubules organizing the centers that we will study when we will do the cell division. Out of the 2 centrioles in a spermatozoon, the distal one gives rise to axial filament or the tail part of the spermatozoon, right? Centrioles can be transformed into basal bodies, right? And the basal bodies formed from centrioles, they give rise to the cilia and the flagella. So the basal body which is formed from the centrioles will give rise to the cilia and the flagella, right? So that's about the centrioles. So we've talked about the cilia, we've talked about the flagella, we've talked about the centrioles, and very briefly we will go through the ribosomes, right? So ribosomes are a structure or cell organelle that is found in a eukaryotic as well as a prokaryotic cell. They are also known as pallid particles. They were discovered by Robinson and Brown in plant cells and by Pallid in animal cell. That is why they are known as pallid particles. So we will move on to the ribosomes now. Right, so ribosomes, 2 types of ribosomes found in the prokaryotic as well as the eukaryotic cells, and they are not membrane bound organelles, right? And 70S type of ribosomes and 80S type of ribosomes are found. So we start with the ribosomes which were discovered by Pallid in the animal cells, right? And because they were discovered by Pallid in the animal cells, they are also known as pallid particles, and in the plant cells they were discovered by Robinson and Brown. So Robinson and Brown, they discovered the ribosomes in the plant cells, right? So a large number of ribosomes, they occur in the cell, right? So they are also known as the protein factories because they carry out the synthesis of proteins, right? They are subspherical in outline, not a complete sphere, subspherical in outline. A covering membrane is absent. That is why we say that it is not a membrane bound organelle. Now if we draw the ribosome, we draw it as one part and we draw another part, right? Subspherical structure, and this region, the space that is there, is for the RNA, to form the protein. They carry out the amino acids over here to form one particular protein, right? So the covering membrane is absent, and that is why it is not called a membrane bound organelle. each ribosome, now if you look at the structure, it consists of two subunits, right? Larger dome shaped here, larger dome shaped, and a smaller oblate or ellipsoid in structure. The larger subunit, you know, there is a particularly it is shaped with a depression so that the smaller subunit fits into that, right? That is how they are arranged. So the smaller subunit, it possesses a platform, a cleft and a head with a base with which it fixes into the larger subunit, right? Ribosomes may occur singly. If they occur singly, then they are known as monosomes, and if they occur in a helical or in a, you know, in a group, they are known as polyribosomes or polysomes, right? Poly means many and soma is body. So the different ribosomes of a polysome, they are all connected to each other, right, by strands of messenger RNA, right? The maintenance of polyribosome requires a lot of energy. So they are formed during the periods of active protein synthesis. The polyribosomes are formed when, you know, very actively the ribosomes are involved in the synthesis of protein or the formation of protein, right? And number of copies of the same polypeptide are required, then only they are found more in number. They occur in all living cells, right, with the exception of mammalian erythrocytes, right? Erythrocytes, red blood cells, they are not found. Depending upon the place of their occurrence, ribosomes are of two types, cytoplasmic and organelle, right? The organelle ribosomes are found in plastids, right, and mitochondria. If you remember, when we talked about the mitochondria and the plastids, we said that they have their own, first of all, they are semi-autonomous structures, and then we said that they have their own DNA and they have their own RNA, right? So this kind of RNA which is found in a particular organelle is known as organelle RNA, and they are 70S type of ribosomes, right? So organelle RNA are 70S type of ribosomes. As discussed, Swedberg is the unit, and that is why it is 70S type, right? So that is the ribosome found in the organelle. Other than that, the cytoplasmic ribosomes or the cytoribosomes that remain in the cytoplasmic matrix, and the cytoplasmic ribosomes are the 80S type of ribosomes, right? So size of the ribosomes, of course, they differ, right? 80S type of ribosomes are synthesized in the nucleolus. Synthesized means where are they formed? They are formed in the nucleolus. Proteins come from the cytoplasm, and that is how they make up the ribosomes, right? What are the function of the ribosomes? They are popularly known as the protein factories. So that means they are the sites of protein synthesis. That means protein synthesis takes place over here. They are free and they are attached ribosomes also. Free ribosomes found in the freely in the cytoplasmic matrix, and attached ribosomes, if you remember, we talked about the endoplasmic reticulum, in which we said that the endoplasmic reticulum is rough endoplasmic reticulum and smooth endoplasmic reticulum. So the rough endoplasmic reticulum has ribosomes attached to it, right? So again there the protein synthesis takes place, right? Then enzymes and factors. Ribosomes provide enzymes also, right, and factors for condensation of amino acid to form a polypeptide chain, right? So those are the functions of ribosomes. Basically the protein factories, and wherever they are found attached to any structure like the endoplasmic reticulum, there also the protein synthesis takes place, and then they help in the formation. They are enzymes and factors. That means they provide, these ribosomes, they provide enzymes for any particular, you know, function, and they provide factors to form a polypeptide chain, right? So that's about the ribosomes. And now we move on to the last part, that is the nucleus, right? Nucleus, a very, very important structure again of the cell, right? That is called the brain of the cell, right? And we know when we talk about the brain controls the whole body, right? So now if nucleus is the brain of the cell, That means nucleus controls many of the activities of the cell. So we start with the nucleus, right. So starting with the nucleus, brain of the cell, and nucleus is found in almost all the cells. It is a cell organelle which was first described by Robert Brown, right. So Robert Brown, an organelle which was first described by Robert Brown, right. It is a specialized double membrane bound protoplasmic body which contains all the genetic information for controlling cellular metabolism, reactions, right, and transmission to the posterity, transmission of those things to the next level, right. So it is a specialized double membrane structure. So if we draw a cell, a nucleus, okay, right. So double membranous structure here, a double membranous structure. Right, and we see here that this is known as the nuclear envelope, right. So this is the nuclear envelope, right. If we see here, the nuclear envelope is not joined at certain places, and these spaces or the pores that are present are microscopic. Here they are visible, right. So just for you to know, right. So these are known as the nuclear pores, right. Now nuclear pore keeps the movement of the contents of the nucleus and the cytoplasm, right. So contents can move out from the cytoplasm, things can come in, right. So that is the nuclear pore, and we see that it is a double membranous structure. A nucleus in the non-dividing or the metabolic phase is called the interphase nucleus. It says here, like other cellular structures, living unstained nucleus that does not show much internal differentiation. You won't be able to find out things inside the nucleus at that time. For detailed study of the nucleus, the cells must be properly killed, fixed and stained. So staining is required to see the internal structure. So nucleus is the largest cell organelle, right. Though first the nucleus was observed by Leeuwenhoek in red blood corpuscles of fish, but it was well described by Robert Brown, right. Then nucleus is present in all the eukaryotic cells with the exception of mature sieve cells of vascular plants, and they are also absent in mature red blood cells, right. Why they are present in only the eukaryotic cells? Because in the prokaryotic cells a well defined nucleus is absent. It is just an entangled mass of DNA which is known as nucleoid that is present in the prokaryotic cells, right. Presence of heredity information in the nucleus was proved by Joachim Hammerling. You need not learn that name, that is just for your information, right. So the nucleus may be present in a cell, a single nucleus, maybe more than one nuclei are present, right. So the cell may be uninucleate if only one nucleus is present. If two are present, they are known as binucleate, and if more than that are present, then they are multinucleate, right, or polynucleate, right. So multinucleate or polynucleate condition is found in some cells of bone marrow, right. So in bone marrow, some cells of bone marrow polynucleate. Bone marrow is polynucleate. Some cells of bone marrow, striated muscles, right, latex vessels, and in several fungi also multinucleate, cells are multinucleate, right? Now, it says here, in plants and fungi, they are known as cenocytic cells, right? So instead of using the term multinucleate, cenocyte we use for plants and fungi, right? Nucleus, if we talk about the position of the nucleus is usually found in the region of maximum metabolic activity in the cytoplasm. That means in the cytoplasm where maximum reactions take place, chemical reactions take place, maximum activity is there the nucleus is found, and it is commonly situated in the geometric center of the cell. Generally in the animal cell, although we know that in the plant cell it is slightly towards the periphery because of the occupation of the central vacuole in the plant cell, right? So it is pushed towards the periphery. Nucleus is peripheral in fat storing cells also, that is the adipose cells, and it is basal in the glandular cells, right? So that is the position, but basically found in the central position, right? So shape also may be rounded, may be elliptical, right? So that is the different shape. Now we come to the ultrastructure, in which I explained the nuclear envelope, right? It bounds the nucleus on the outside. The nuclear envelope, it separates the nucleus from the cytoplasm. Outside the nucleus is the cytoplasm. So the nuclear membrane separates the nucleus from the cytoplasm, and it is made up of two lipoprotein trilaminar. We talked about the biomembranes, or we talked about the cell membranes. They are made up of lipids and proteins. It's trilaminar, so both the membranes are the same, right? Then the inner membrane is smooth, the outer membrane may be smooth, or maybe cytoplasmic surface may bear some ribosomes like the endoplasmic reticulum, right? So that means ribosomes may be attached to the outer membrane of the nuclear envelope, but inner membrane is all smooth without the ribosomes, right? So that is the nuclear membrane. If you look at the nuclear membrane, in between the nuclear membrane there is space, and this space is known as the perinuclear space, right? That is the perinuclear space. Now the nuclear envelope, it contains large number of pores. We have discussed, right, the nuclear pores that are present. These pores are the perforations, right? Now inside the nuclear envelope is the matrix, right? And the matrix is known as the nucleoplasm, right? It is a transparent, semi-fluid and colloidal substance which fills the nucleus. So that means everything, all this whole thing is the nucleoplasm. Like the cytoplasm, it is the nucleoplasm, right? It contains the nucleosides, number of enzymes, right, related to the formation of DNA, the RNA, etc. Some nucleoproteins are present in the nucleoplasm, right? Then other than the presence of the nucleoplasm, there is a network of fine fibrils also that are present, which are made up of acid proteins, and they function as the chromatin, right? So you know, chromatin network is also found in the nucleoplasm. There will be whole network of chromatin fibers like this, right? So that is that we can refer to as the nuclear matrix, or we will label is as chromatin fibers, right? They are the chromatin fibers, right? Now other than the chromatin fibers, this is, what is this chromatin? It is the hereditary material, the DNA, right? Protein fibrillar complex is there, that means proteins are attached to this, the histone proteins are attached to the DNA, and it is named so because of the ability to get stained with certain basic dyes, right? Chroma is color, that is why they are known as chromatin, right? They can take up the stain or the color. Chroma means color. That is why chromatin. Now they occur in the form of fine overlapping coiled fibers. If you look at these are all coiled and they are overlapping each other, right? Like coiled fibers, and they produce a network like appearance, right? So this is also known as chromatin fibers or chromatin reticulum. When we use the word network, we use the term reticulum. Right? Why reticulum? Because these fibers are all coiled, they are overlapping each other and they are forming a network, and so they are chromatin reticulum. Right? Then inside this we also find the nucleolus. Right? Nucleolus is a structure which is found inside the nucleus, right? And it is a naked, round or slightly irregular structure which is attached to the chromatin at specific regions, right? So it is just an aggregation of structure. The nucleolus, let's say this is the nucleolus or the structure that is also present inside the nucleus. Nucleolus, right? Nucleoli is the plural term, right? A covering membrane is absent in the nucleolus. Maybe more than one nucleolus may also be present, then it is the nucleoli, right? And it says here in up to 1600 nuclei reported in the oocytes of Xenopus, right? So one nucleolus or maybe more than that. A covering membrane is absent around the nucleolus. Calcium seems to be essential for maintaining its configuration, right? It has a lot of things inside. There are some RNA granules, there are proteins, etc. That are found inside the nucleolus, right? So it is a part, the chromatin portion, it is a part of the chromatin which is associated to the nucleolus. Now depending upon its position, the nucleolar chromatin is of two types, perinuclear and intranucleolar, right? So nucleolus is the principal site for the development of ribosomal RNA. Right, so RNA forms inside the nucleolus. It is the center for the formation of ribosomal components also. It stores nucleoproteins and the same are synthesized in the cytoplasm and transferred to the nucleolus. It is essential for spindle fiber formation. So what all happens inside the nucleolus? That the RNA, mRNA, ribosomal RNA is formed inside the nucleolus. It is the center for ribosome components. That means what makes up the ribosome, right? Nucleolus stores nucleoproteins, proteins found in the nucleus, right? So nucleoproteins. If we come to the function of the nucleus, we see that it is an essential and a very integral part of the eukaryotic cells. Why? Because it stores the information in its DNA molecules, right? And this DNA can be passed on to the daughter cells. So, and other than that, it also controls all the metabolic activities, right? Chromatin, this is the chromatin in the nucleus, contains the hereditary material, right? So this is the chromatin and this is the hereditary material. Right? Chromatin is DNA protein complex, right? That means DNA associated with proteins, right? There are different stages of the cell. So at one stage, mostly you will find the DNA to form a network inside the nucleus. Other than that, at certain stages at the time of cell division, you will find the chromosomes inside. Right, so the chromatin, the material, it gets arranged into the chromosome at a specific time during the cell division, right? So that is the DNA, right? Then the nucleus also stores the genetic information. Since chromatin is a part of the nucleus, it possesses all the genetic information that is required for the growth and development of the organism, right? This chromatin is actually the DNA. And DNA stores all the genetic information. Then nucleus, it controls the cell metabolism and other activities of the cell. Formation of the RNA is also controlled by the nucleus. Only when the nucleus gives the direction, right, to start the formation of the RNA, then only the RNA would be formed, right? So it controls the synthesis of particular type of enzymes also. Then ribosomes, as I told you, nucleus is found, nucleolus is found inside the nucleus, and nucleolus, inside that ribosomes are formed. So ribosomes are formed in the nucleolus part of the nucleus. Then variations or changes are also associated with the genetic material. Cell growth and maintenance of the cell with the help of RNA. The nucleus directs the cytoplasm, the synthesis of specific structural proteins also, right? So replication is also an essential part, and replication of nucleus is essential for cell replication. So that is also controlled by the nucleus. The nucleus, right? Now we come to the next part, which are the chromosomes. And when we talk about the chromosomes are the, they are found in the nucleus in the interphase, part of the cell division of the cell, right? So I told you that when we talk about the nucleus, there are various stages, right? In one stage you will find it to be in a network form, the chromatin is arranged in a network form, and at one stage you will find the presence of chromosomes, right? So in the interphase, this is one phase of the cell division. We find it says here, it has a loose and indistinct network of nucleoprotein fibers which are called the chromatin. So this is the interphase stage of the cell division. But during the different stages, as I told you, of the cell division, the cells show structured chromosomes, right? So structured chromosomes are also found, and these, as I told you, chromatin contains DNA, and some proteins which are known as histone proteins are also associated. It is around the DNA. If we talk about the length of the DNA, it says a single human cell has approximately 2-meter long thread of DNA, 2-meter long thread of DNA, which is distributed in its 46 chromosomes or 23 pairs of chromosomes. 23 pairs of chromosomes are found in a single diploid cell in the nucleus, right? Chromosomes are found in the nucleus. So that 2-meter long DNA is found in all the 46 chromosomes, right? And if we, the detailed of the DNA structure you will study in class 12, and we'll just very quickly go through the structure of the chromosome, right? So now if we talk about the structure of the chromosome, and you will follow it in your books also. So they are, a chromosome, if we look at a chromosome, basically this is the structure of a chromosome, and we find a spherical structure attached to the chromosome, right? So now if we look at the chromosome, this spherical structure is known as the kinetochore, right? Every chromosome essentially has a primary constriction depression, which is the centromere, on the sides of which a disc shaped structure known as the kinetochore is present, right? So this depression that is present here is the centromere. Yeah, this is the kinetochore, that is present, right? Now depending upon the position of the centromere, the chromosomes are named differently, right? Now if this centromere or this constriction is present at different places, as I told you, if the centromere is right in the terminal end, if this is here, right, and these are the two strands of the chromosome, chromatids, right, this is known as the telomere, right, depending upon the position of the centromere. Now if the centromere is inner to the telomere, slightly like this, right, then this is called acrocentric, right? Then submetacentric, in which the centromere is not exactly in the center, so one part of the chromatid is longer than the other part of the chromatid, right? And then comes the metacentric. This is the metacentric, where exactly the centromere is present in the central part of the chromosome, right? So which divides the chromatids into upper and lower equal length parts. So that is metacentric, right? So that's about the chromosomes, and we've completed the lesson, the cell. Finish your assignments, post it to me. I'm still not getting all the assignments, and we will start with the next lesson. We will talk about the animal kingdom in the next class. Please complete your work and post your assignments. Thank you.